Hey YouTube, just going to do a series of videos suggested by I Live On, which is about genetics and genetic mutations. But I figured that, seeing as this will be aimed at teaching creationists basic science on genetics, rather than teaching people who have actually had the basic science background, I'll start with the basics, like you would for a child. So one of the first questions is usually asked by a child is, why is DNA described as a double helix of life? And well, the double is from the two strands of DNA. A helix is anything spiral in its shape or structure, and DNA is bound in two spiral strands. Without the DNA there would be no life. So I'll draw a double helix here. Okay. And I'll cover on what the blue, red, yellow and green bits are later on, but that's a basic double helix structure for DNA. Okay, another one is where are chromosomes found in the living cell, and I'll show you that by drawing it. Okay, so you get your basic cell structure. Alright, I'm not going to draw the entire thing, and you get your nucleus of the cell here. It's a pretty big nucleus, but usually shaded black in most diagrams, okay? And you get your chromosomes, two base pairs there, see? Whoa, that's useless from afar. Okay, and that's where you'd find them. Okay, so you'd find them. Humans generally have 26 base pairs, I believe. I'd have to double check, it's been a while since I last done it. So I'll draw you a chromosome, enlarged. And that's a chromosome. And generally they'll have loads of these strands inside them. Okay. So that's two basics questions covered. And now, what else am I going to cover? Well, I suppose we could cover what genes are. And inside every cell of each living thing, plant or animal, are sets of instructions called genes. The genes provide the instructions on what the plant is or what the animal is, what it looks like, how it's to survive, and how it will interact with its surrounding environment. The genes are strung together in long strands of material called deoxyribonucleic acid, which is DNA. And these long strands are called chromosomes, like I've shown you. Okay. Most living things have pairs of chromosomes, as in one from each parent. Though they may have different number of chromosomes from another living thing. For example, that's 23 pairs humans have. Memory's old, it's been 12 years since I studied this. And the fruit fly has 4 pairs. Each gene is made up of long combinations of 4 different nucleotide bases. Remember folks, that's the different coloured bits. Okay. And these various combinations of nucleotide bases that determine everything about a living creature. The 4 nucleotides are called adenine, which is A, cytosine, which is C, guanine, which is G, and thymine, or thymine, depending on your pronunciation, which is T. The genes for green eyes might have a nucleotide sequence AAA, CC, GG, TT, TTT. And the gene for blue eyes might have this nucleotide sequence AAA, CC, GG, TTT, AA. I'm going to draw them out for you. Give me a second to sort this out. Now you'll notice here the code for green eyes and the code for blue eyes are not very different. You've got the 3 A's at the start, 2 C's, 2 G's, 3 T's, and then it's only the 2 last nucleotides here that change, the colour-tide sequence. Getting TT for green eyes and AA for blue eyes. So it's really the last 2 characteristics here that state where you're going to have blue eyes or green eyes. The structure would be the same. Okay. And it's not an accurate gene formula, by the way. Correct sequence would be about several thousand bases long. That's just a diagram for you. Many genes have a quality known as dominant or recessive genes, And dominant traits are more common than recessive traits due to this quality. I'll show you a basic structure for dominant and recessive genes. Okay, excuse me. I'm going to draw you a little diagram here, basing it on dominant and recessive genes of the eye colour. We're going to use brown and blue for this example. So, brown is the dominant eye colour in genetics. Okay, and blue is the recessive. And to show that in diagrams, okay, that's the brown there. Oh Christ, this really is bad when I try to focus on drawings, isn't it? The dominant gene, and that's described as a capital B. And the blue, being a recessive gene, is described as being a lowercase g. Now that's always the case in genetics. The dominant will be the capital letter and the recessive will be the smaller letter. Okay, so looking at this diagram here, I know it looks confusing, but here's the mother. So everyone has two symptoms of two genes, the dominant and recessive trait. Now, presuming that both parents have brown eyes, because they've got the dominant and recessive gene with both the dominant and recessive gene, BB, that would split off like this. You'd have the B, the dominant gene set here, the recessive gene here. So that would split into two categories here for recessive trait. You'd have the lowercase b, lowercase b. The dominant would be here and here. And from the father's side, it would of course split, with dominant being here. So if dominant goes and meets this one here, then it's going to be brown-eyed because it's got the dominant gene here, which dictates whether it's going to be brown-eyed, whether the child's going to be brown-eyed. Whereas if you take the recessive gene and it meets the recessive gene from the mother, it's going to be blue-eyed. However, if it meets the father's recessive gene meets the dominant gene of the mother, it's going to be brown-eyed. And if both the dominant genes meet, then the baby's going to be brown-eyed. And there we go. And it's more likely that the child of this pairing would be, of this one up here, would have brown eyes. The child of this child would have brown eyes, because that's the dominant trait there. So there's a 1 in 4 chance of the baby having blue eyes. Christ, that's a confusing chart. I could have done that better. So say one brown-eyed parent had the recessive gene, okay, and was completely recessive. Well, that changes it. Hold on, I'll draw that. So as you can see, I've changed it now to show the mother still having a dominant gene, but the father having two recessive genes. So that splits off into here. So you've got the recessive gene here, meeting the recessive gene of the mother. And again, you've got the second recessive gene from the father meeting a recessive gene again from the mother, and this creating creating two brown-eyed children. Then you've got the dominant gene here meeting with the recessive of the father, and again the same, meaning that's going to be a brown-eyed child. So you got a 2 in 4 chance, or rather a 1 in 2 if you're going to half it down a bit. Okay. Now, if you get a brown-eyed parent without the recessive gene, then I'll show you what happens there. Okay, so I've changed it again, so the mother has fully dominant genes. She's got both genes as dominant brown gene, and the father still has the 2 recessive blue genes. So this is what happens here. You'd get the dominant gene meeting a recessive gene in every single case. So it would be 100% brown-eyed child. Okay. And it doesn't matter who has the recessive and who has the dominant, the results would still be the same. Okay. I hope that's clear. If it's not, let me know. Just want to get a quick word definition out. Dominant. By dominant gene I mean a gene in one strand of DNA that is stronger than the corresponding gene in the other strand of DNA. And by recessive, it's obviously a gene in one strand of DNA that is weaker than the corresponding gene in the other DNA strand. And a trait is a distinguishing feature in a person. Just getting that out there. So, now you've had my basic explanation of what a double helix is, how dominant and recessive genes work, and where chromosomes are found and what a chromosome is, which is of course a strand of DNA, and what the nucleotide bases are. I'm going to try and show you what DNA replication and protein synthesis do. Now, I'll go over the basics. Knowledge of the structure of DNA began with the discovery of nucleic acids in 1869. That genes control the synthesis of enzymes was understood in the 1940s. In 1953, an accurate model of the DNA molecule was presented thanks to the work of Rosalind Franklin, James Watson and Francis Crick. Now, I'll see what I can do for you folks. Okay, so back to the nucleotides bases, okay? And what we want to do is, you notice that each base has an end to it. That's very unique. Okay, so in general you'll find that A and T go together because the T has, well, in this demonstration, the T has this shaped end and the A has the shape indent that can accept that. Whereas if it was to try and match with G, it wouldn't work because the G has a wavy end here, which works with C because C has a wavy end too. So generally A and T, C and G are the pairs that stick together. Okay? And that's how the DNA is. I'll show you a basic longer strand of them connected. Now the first step to DNA replication is to unzip the strand like this. Ignore this for now. Unzip the strand like this, okay? Now in a real cell, the molecule unwinds from spools made of protein, then untwists, because you've already seen the double helix where it's all twisted up like that. Well, here I've already unwound and untwisted the molecule for a demonstration. The DNA will now resemble a ladder, like this one, and enzymes, which are special kinds of proteins, move up the ladder and breaking the rungs apart like this. Okay, now to make a DNA strand, you'd have to match up the pieces. Now, in a cell, that would be a case of, say, that moving to meet that, or that could move up to meet that, or such like. Okay, things would move around. Now I don't have a full demonstration here I don't have one I can move around to show you, obviously. So the base pairs would move, the nucleotide bases would move around to match up with their corresponding base pair. Now, adenine complements thymine, and guanine complements cytosine. Cytosine even. Okay. Now, when they're put together, it would be like this. This is how it would seem when they're all together. Okay. And that is what happens. That's replication there. Our new DNA molecules consist of only a few base pairs here, but that's not so with real DNA. Real DNA, each human chromosome, for example, contains millions of these base pairs. All 46 human chromosomes contain 3 billion pairs. So next we're going to cover protein synthesis. Where are these strands found? Well, I kind of already covered that. They are found in a cell. Now I'm going to do it like this time. Okay, and you got loads of wee dots all over the place. Right, now the wee dots that you see there, well, they're called ribosomes. Ah, hell, hold on. And I'm not going to cover the cell structure just now. But this, of course, is the nucleus. This big bit here. And inside that is where you find the chromosomes. Okay? Bearing in mind, I'm trying to keep this at a child's level. So if it's too complicated, please let me know in the comments below. And I'll try and do another one where it's simplified. Okay, now, protein synthesis. Well, just like with replication, this is a basic start of protein synthesis. The splitting up of all the base pairs. The base nucleotide pairs. Okay? And like DNA, RNA is made of 4 bases. 3 of these bases, adenine, cytosine and guanine, are the same as DNA. But instead of thymine, which is T of course, the 4th base is uracil. Hold on. Or U in our future demonstrations. And each base is a complement. Another base that it can connect to. So this time round, A connects to U, and as in DNA, C connects to G. Now RNA is ribonucleic acid. Okay, and mRNA I presume means molecular. Well, no, mRNA means messenger RNA. Okay. So, now, so what we'd have, okay, we've got a wee diagram here. You can see the big purple thing is a ribosome. The mRNA strand is at the top. Okay, and you've got something here which is nucleus. Now, here's a quick description of what's happening here, okay? A ribosome will attach itself to the mRNA strand. Ribosomes are a cell structure for building proteins. mRNA bases are grouped into sets of 3, called codons. Much like this, okay? Each codon has a complementary set of bases called an anticodon. This one here. Anticodons are a part of the transfer RNA, or tRNA molecules, which is this thing here. Attached to each tRNA molecule is an amino acid, and in this case it's glycine, okay? Right. So that's the first codon matched to its first anticodon. What happens next is that the ribosome will move again to the right, leaving this 3 codons here free to be matched up. So what's happening then is that the next complementary anticodon will come along and attach, dragging through its amino acid as well in the same process. Okay, it won't be glycine, but you know, it'll be the next one to attach there. And what will happen then is it will bind the amino acid will bind to the glycine amino acid. And after that, the ribosome will move to the right again, and the first tRNA will float away from the ribosome to pick up, leaving its amino acid behind. And it will pick up another amino acid molecule, ready to wait until it's needed again to transfer in the next one. So what? Okay. And then after it's moved to the right for the 3rd time, okay, so it's got this one, it's here now, it can move away to the right, and that will become 2 amino acids linked together. Now it'll move away again, and this is where this bad boy comes in to link up again. So and this will carry on until a ribosome disappears. Okay, and this leaves behind what we call a polypeptide chain. And in this case, our diagram's case, it'll be just 3 amino acids. And what will happen is, with a polypeptide chain which is the protein polypeptide chain is a protein under construction, by the way. More and more will be added until the chain is about 100 to 10,000 amino acids long. And the protein synthesis will end when a stop codon is reached. And the ribosome will then release the polypeptide chain. And that is how protein synthesis happens. I hope this. Now, I hope I was clear enough. Bearing in mind it's been 10 years since I left school, so it's been about at least 12 years since I last studied this stuff. 11 years since I last studied this stuff. And this is a refresher course for me as well. But I'm hoping I'm explaining it well enough. And if I'm not, then I'll take the video down and re-edit it. Not re-edit it, sorry, redo it so I can explain it better. I'd like some feedback on this, please, so do leave a comment down below if you watch this. And I hope it was good enough for you.